Hi guys, this is Caroline with Fiveable, and I'm super excited to be streaming with you tonight. And as always, follow us on all sorts of fun platforms. Okay, so today we're going to talk about what heredity is. We're definitely going to talk about some key vocabulary because that's probably the most important thing with all of this. This is a very vocab-heavy section. We'll talk about both Mendelian and non-Mendelian genetics with some examples, and then we'll do some chi-square. All right. So a little background. That lovely strapping gentleman on the left is Mr. Gregor Mendel. I'm sure you've all heard of him. He was a monk. He lived a long, long time ago. And he is the father of modern day genetics. So he's a great guy. We learned a lot from him and his pea plants because he was busy as a little, I was going to say farmer, gardener at his monastery. And he started to notice these different characteristics in his pea plants. And then when he would cross them, he would see all these different phenotypes, as you can see. So I'm sure you've heard of Mendel and his peas. Super important dude. And some vocabulary here for you while we're on this. So what you see here are a couple of different traits and then different phenotypes within those traits. Okay, so flower color is a trait, and then within that you can have white flowers or you can have violet or red flowers. Okay, so those are the two different phenotypes that are going to be controlled here. So phenotype is the physical appearance, and then we'll talk about genotype as well, which is the genes. So this was his first kind of delve into this. He looked at 7 different traits with these pea plants. Funny enough, crazy enough, Mendel did not know that DNA was our genetic material. That's actually a relatively new discovery, which is totally crazy for students to think about and me to think about, is that we didn't really know until, within the past like 80 years that it was DNA that was our genetic material. So Gregor Mendel had no idea that it was DNA that was being passed on in his pea plants. He just knew that something was being passed on. They actually thought it was protein for a while just because there's more amino acids than there are nucleotides. And then after Mendel as well, we discovered these things called alleles. So alleles are going to become really important. They're just different versions of a gene. So you can see that here. We have a gene. We have allele 1 and allele 2. They're in the exact same location because they're the same gene, okay? But they're just slightly different. You can see one has more blue and one has more like greenish yellow. So that would be signifying some different nucleotide pairs that are going to make a different phenotype or physical appearance, and we'll talk more about that as we go. But again, just some vocab that I'm sure you've heard many, many times, but just a little refresher, never hurt anybody. Okay, so the basis of genetics is this idea of genotype and phenotype, as I've been talking about. There's a Punnett square there. We'll do a couple little basic practices, but again, I'm sure you had that in like an intro to bio class, so I won't spend too much time on it. But you do need to know that phenotype is the physical appearance and genotype is the genetic component of that. So we have these two parents that are both heterozygous because they have one dominant and one recessive allele. We could also call them a carrier for that. And something that's important to remember that they oftentimes touch upon on the AP exam is what it really means when you have these dominant or recessive genes. So remember that genes just code for proteins. So usually, let's say purple is dominant to white. If you have the dominant allele, you make a certain protein that creates this purple pigment. And if you don't have that allele, then you don't. So if you're heterozygous, if you have one dominant and one recessive version, you're still producing enough protein to produce that purple pigment to appear purple, because we know heterozygotes appear the dominant phenotype. And we can see that here. Both of these, why is this so big? What is going on? Okay. Both of these guys are heterozygous. We can see they have one recessive and one dominant allele, but they're appearing the dominant color. That's because they have enough of the protein of whatever it is to produce that dominant phenotype. So again, if it's the purple pigment, they have enough of that protein producing the purple pigment to appear purple. If you have neither of those genes, then you're not making any of that protein that would be producing purple pigment. This is like a tongue twister. So instead, you might appear white if that's the purple versus white flower example. So just remember what this really means. It's not just like capital A and lowercase a or capital B and lowercase b or whatever it is that you're writing out. It's actually a gene, a strip of DNA that has a code on it that makes the protein that then contributes to some sort of phenotype. So that's just like the AP version of basic genetics, right? So that's like APifying what you've probably learned for a long, long time. Okay. So then within genotype, you can be homozygous dominant, heterozygous, homozygous recessive. Remember the prefix homo means same and hetero means different. So if you're homozygous, you have two of the same alleles, either two dominant ones or two recessive ones. So these two guys are homozygous, homozygous dominant, homozygous recessive. And then we've already stated these two are heterozygous because they have a mix, they're a carrier, they can pass it on to their offspring, as do these parents, right? 1 of 4 offspring would be expected to have the recessive phenotype, but they themselves are dominant. Good so far? So it's probably all basic review, but it's good practice. Okay, perfect. All right, so in order to figure out the results of a parental cross, we use these Punnett squares. These are very, very simple. Things like dihybrid crosses don't really show up on the AP exam anymore. It's not a bad idea to be familiar with them. I'm not going to go into detail with them tonight just because we're covering so much content tonight, but you can certainly take a look and practice with them. Same general idea. But in order to figure out the results of parental crosses, we really need to know about independent assortment. So independent assortment is just one of many ways that we get genetic diversity through meiosis. So we have crossing over, we have independent assortment, we have random fertilization, all of these things that contribute to genetic diversity, which is the key to life. And I actually just wrote a study guide for this unit that talks all about these 3 things in detail. So be on the lookout for that. That's going to be posted by Fiveable, hopefully sometime soon. But what you can see here, so independent assortment is really nothing other than there is a chance, a 50-50 chance. So let me get my pointer out, sorry. There's a 50% chance that this blue chromosome, so picture the blue chromosome, I'm sorry, let me back up for a second. This is a pair of homologous chromosomes. So this is kind of where like meiosis vocab kind of gets out of control. But these are homologous chromosomes. So we know that as humans, we have 46 chromosomes, but we really have 23 pairs of chromosomes, right? We have 2 versions of chromosome 1 through chromosome 22, and then we have an X and an X or an X and a Y that rounds out, okay? So we have 2 versions of chromosome 1, one from mom, one from dad. 2 versions of chromosome 2, one from mom, one from dad, blah, 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 forever. Okay, so here, these homologous chromosomes, one version of chromosome 1 and another version of chromosome 1, line up to be split in the first process of meiosis, okay? So there is a 50% chance that this blue chromosome from dad will end up on the left. But there's also a 50% chance that this red chromosome from mom will end up on the left, right? It's like flipping a coin, okay? There's a 50% chance that just by the random nature of the universe, one will end up on one side versus ending up on the other side. And you're like, so what? Who cares? But if you look at all of these options, so with these 3 chromosomes, there's actually 8 different ways that these chromosomes can line up. Each of these cells is a different way that these chromosomes can line up. And they're all going to lead to, based on crossing over and based on how they separate, different possible gametes or egg or sperm that can then be combined with another individual's in order to form a zygote, okay? So these guys will separate, and there's a 50% chance that we're going to get the version from mom, and there's a 50% chance that we're going to get the version from dad for each and every chromosome. That's what independent assortment is. That was Gregor Mendel. It does show up a lot. Alleles are, so Madison, great question. Alleles are just versions of genes. So alleles are just versions of genes, so they're going to be definitely on the homologous chromosomes. So with homologous chromosomes, let's take chromosome 1. Let's say for whatever species this is, eye color is located on chromosome 1. So on the left-hand side, we have the version of eye color from dad, and on the right-hand side, we have the version of eye color from mom. So maybe dad is dominant for eye color, and so he has a dominant allele, and then mom has a recessive allele. So there's a 50% chance that this offspring is going to inherit the dominant allele from dad, and there's a 50% chance that this offspring is going to inherit the recessive allele from mom. Okay, so remember that's just a version of a gene. Alleles are just DNA, so they do exist on the homologous chromosomes. Hopefully that answers that question. Absolutely. That's what I'm here for. So keep asking. So this all sounds kind of confusing and it leads to a ton of variation. So actually, because it's a 50% chance, you can take, it's 2 to the nth power, where n is the number of unique chromosomes that an organism has. And that's how you can see how many different combinations can be made. So this is 2 to the third because they have 3 sets of homologous chromosomes, or 8 possibilities. If you do 2 to the 23rd, it's over 8 million possibilities. So each individual human, without crossing over, just from independent assortment, can make 8 million possible egg or sperm just from this process of how these chromosomes line up. Pretty crazy. Much more simply, because this can get pretty complicated and it's more of a meiosis topic anyway, we do this, we practice this all the time when we're making a Punnett square, which you guys have probably been doing for forever. So in this example, both of our parents are heterozygous. Okay, and what you do when you're doing a Punnett square is you separate the dominant allele and the recessive allele, and they're each on top of their own column or at the end of their own row. And what that's showing is that there's a 50% chance that you're getting this allele versus a 50% chance that you're getting the other allele, which is the whole point of independent assortment, where those things are being separated. Okay. So you show this all the time when you're making a Punnett square, which is how we figure out these probabilities. But that's the kind of the science behind it. And independent assortment is definitely a really important AP concept. And again, I did just make a unit review guide that Fiveable will be putting out that goes into more detail with that too. Okay, so we've separated the 2 parental chromosomes, okay, the 2 alleles, the dominant and the recessive. In this case, both parents are heterozygous, so they both have a dominant and a recessive, okay? And then we're combining them row to column. I used to teach my freshmen that this was like battleship, right? You have this allele and this allele coming together in this location, and they're making a homozygous dominant offspring, and it was all dramatic, but you guys get the gist of this. So we fill it in, and then we have our probabilities. So there's a 75% chance that this offspring will have whatever the dominant trait is, and a 25% chance that they will have the recessive phenotype, which is not the same as the genotype breakdown, right? So it's a 3-to-1 ratio for dominant to recessive phenotype, and then it would be a 1 to 2-to-1 ratio of homozygous dominant to heterozygous to homozygous recessive. So there's a 50% chance that the offspring here will be heterozygous, just like the parents. Okay, again, figuring this is mainly review, but are there any questions? Okay, perfect. Oh, forgot that I wrote this all out for you. It's like my sick brain. I don't know. It's just not all of my blood is flowing to my brain. But there you can see, and you have it in front of you, all the different ratios there. Okay, so just really quickly, if we have an individual that's heterozygous versus an individual that's homozygous dominant. All right, we're going to go to bit paper if you're not familiar. Okay, I'm going to add a new page here, start out fresh. Hopefully you guys can see this. So Punnett square, we had a heterozygous individual and a homozygous recessive individual. So as we fill this out, just take a second to look at this, and I want you to just tell me what your genotypic versus phenotypic frequencies would be, and then I'll go through them with you. But I'll give you about 15 seconds to think about it yourself. Okay, so our phenotypic ratio would be 1-to-1, right? There's a 50% chance that this individual is going to have the dominant phenotype, and a 50% chance that this guy's going to have a recessive phenotype. It's also technically 0 to 1-to-1 for the genotypic ratio, or 0 to 2-to-2, doesn't matter. You can simplify that because these individuals are 50% chance going to be heterozygous and 50% chance homozygous recessive, which is what Natalie just told us. So thank you, Natalie, for answering that. Hopefully that's easy enough. Again, just kind of refreshing. I have seen them ask for frequencies in the past, so it's always good to keep those kinds of things in mind. We're going to go back to bit paper in a second. when we talk about chi-squared and when we look at some of these non-Mendelian traits. But for now, we'll bounce back into what we were talking about. All right, so this is all well and good, and these are probably things that you've learned about for a really, really long time. You have dominant, you have recessive, you have these 3 different genotypes, 2 different phenotypes. But unfortunately, someone once told me that science is just, or learning science is just getting lied to less and less each year. It's kind of true. You just learn that like everything is actually a lot more complicated than what you were once taught as you learn more and more. So non-Mendelian genetics, these are all the things that break the rules that we just talked about and that we've probably learned about for our whole lives. So the unfortunate reality is that almost every trait is not inherited in the traditional Mendelian way, especially not in humans. We have very few traits that are just straight dominant recessive. So everything that we've looked at so far has only 2 alleles with a very predictable inheritance pattern. But there are a lot of other ways that things can be inherited. So not too challenging is codominance and incomplete dominance. And then it gets a little trickier when we talk about multiple alleles and sex-linked traits. So we'll talk about those as well. All right, so codominance. When you think of codominance, I want you to think of like a co-captain or a co-teacher. It means together or both, right? So co is like, or a co-pilot. They're both flying the plane, right? They're working together. All examples of that prefix. So if alleles are co-dominant, then both alleles are dominant, okay? So the brown and white cow would be an example of that. Ooh, I changed some of this example and not all of it. My apologies. It should say if color or coat color is codominant in cows, describe what the offspring would be of a brown cow crossed with a white cow. Because this also happens in feather color with chickens, and you can see that as well. But so co together. So when we're talking about these 2 next traits of codominance and incomplete dominance, the only difference is in the heterozygotes of these individuals. So you're still going to have like your homozygous individuals that are just going to be the same as if it was homozygous dominant and recessive. So if you're homozygous for this white allele, you're going to be white. And if you're homozygous for this brown allele, you're going to be brown. Okay, that does not change. That's the same as it would be if we were in typical Mendelian genetics. What changes is the heterozygote. So when these 2 alleles are together for an individual, okay, then we see them both show up. Okay, because think of dominance. Before, when something was dominant, it would show up in the heterozygote individual, okay? So here, both are dominant, so both show up in the heterozygote individual, and we have this lovely spotted cow as an example. So as I said, the other example oftentimes used is with chickens. So there are black and white chickens that when they're mated actually create this really awesome checkered chicken that has both black and white feathers throughout. It's really, really cool. But neither one is overpowering the other. They're both showing up in the heterozygote offspring. Okay, so that is codominance. Just be familiar with it. They will tell you if something is codominant, okay? So it's not like you have to be able to really determine or come up with that on your own if you're trying to do a cross. If they tell you that these are the results, then you should be able to interpret that codominance is occurring. And Madison, that is a great question. It depends on the breed of cow. So these are called roan cows, if you've heard of them. And actually most of them are that red color. But like a typical black and white spotted cow would be a different breed. And I believe that's just the way that they all look, kind of like how zebras all have stripes. But that's a really good question. So this is just a specific breed of cow called the roan. Okay, so then incomplete dominance. So if we think of codominance means they're both dominant. Incomplete dominance means that neither allele is dominant. So what happens then? Well, neither really shows up then in the heterozygote. So with codominance, they both showed up in the heterozygote. With incomplete dominance, neither of them really show up in the heterozygote. And they love to show this with the classic example of these flower colors. So we have a red flower, we have a white flower, and when they cross, they make a pink flower. And again, this isn't all flower types. This isn't how all inheritance works for flowers. It's just this particular breed of flower or species of flower that has this incomplete dominance. So again, you're not seeing red or white in the in-between. You're seeing a mix of both. So neither is truly dominant. And I don't like that, like all the images that I find, and whenever I see this in the textbook, they always include these uppercase and lowercase letters, as opposed to in codominance where they each have their own, which you can see down here, RW and then RW. Here they still have the dominant or the uppercase and lowercase letter. I'm not sure why. I don't really like it because red isn't dominant, as you can see by this pink in between. But for whatever reason, that seems to be how they write it. So again, those two, the difference really is just with the heterozygote, you still have both extremes on either end. It's just how they manifest in that in-between. And it's kind of like who wins out. And sometimes it's both and sometimes it's neither. Any other questions with incomplete dominance? All right, so multiple alleles is how a lot of our genes or our traits work. Multiple alleles and then multiple genes, which we're not going to get into because it gets real complicated. That's where like multiple different areas of DNA are controlling the same phenotype. Things like skin color would be an example of that. But multiple alleles just means that the genes have more than just a dominant and a recessive. They have more than just two alleles that are controlling them. So with this, they usually write the alleles in the upper right-hand corner of some base letter because there's more than just an uppercase and a lowercase. We actually have multiple different versions of these genes. So in this example here, we have a dominant capital C, and C's are hard, but that's what they use. This is for Himalayan rabbits, okay? And they have all these different colorings. They can be albino, Himalayan, which is this like black and white, really cool looking, chinchilla. And then the wild type is this brown fur. So they have this dominant allele, and then they have these three different recessive alleles. They have the recessive allele CH for chinchilla, the recessive allele H for Himalayan, and then the recessive allele just a lowercase c, which produces no pigmentation, and that would produce a white rabbit. So with this, we have these four different phenotypes this time instead of just two. Then the other thing is we have this dominant allele, and then we have three different versions of recessive alleles, okay? But because this is dominant, it's the same rules apply. If they have one version of the dominant allele and then they have one of these recessive alleles, they will still have brown fur because this allele is dominant and therefore this pigmentation will be produced. Okay. In order to have these three funky genotypes, they need to be homozygous recessive for that specific genotype. Okay. And then when we get into mixing all of these, I have no idea what happens. So we won't get into it because I'm not sure. We'd have to ask some sort of rabbit breeder. So that's kind of what's going on there. Let me see. It looks like someone asked, oh, Madison, how would the genes code for pink specifically? Okay, so Madison, what your question is asking, we have to think back. Sorry, Madison asked, how would the genes code for pink specifically in incomplete dominance? So what happens there is remember that genes code for a protein that codes for a pigment. So both genes would just be producing a little bit of pigment. Okay, so we'd be producing a little bit of white pigment from one protein and we'd be producing a little bit of red pigment from the other because they're both there and they're both producing a little bit of pigment. We see that mix. It's literally like mixing paint colors. So we have a little bit of white pigment and a little bit of red pigment being produced in order to produce that pinkish color. So we wouldn't see this, to kind of clarify, we wouldn't see a red and a white flower make a blue intermediate, right? We're only seeing that pink because it's a mix of the two extremes. It's a mix of those two pigments. So hopefully that answers that question. All right. So the other, my absolute favorite example of multiple alleles is with blood typing. So whether you know this or not, we have the same kind of situation of blood types, and this is French. That's why it says "ooh," that means "or" in French. It's a French picture. We have the same type of situation with our own blood types. So we have A alleles and B alleles, both of which are dominant. Then we also have O alleles, which are recessive. So what you will see here is if you have a blood type, and this is without the pluses and the minuses, that's a different factor, and we're not going to get into that. But if you have a blood type, you might have two dominant A alleles, or you might have a dominant A allele and a recessive O allele. That's what I have, because my mom is blood type O, and I'll get into that in a second, but I have type A blood. So I know that I have one O from her, and then this dominant A, which is giving me A type blood. Same with B. You can have two B alleles, or you can have a B allele and an O allele, okay, which is recessive, so you would still have B type blood. If you have a B type blood, which is the rarest kind of blood type, it's because you have an A allele and a B allele. Both are dominant, so this is an example of codominance. They're both showing up, okay? And then the last is type O blood, which is actually the most common, type O followed by type A. And that is two recessive alleles. So O is a recessive blood type. So A and B are codominant, and O is recessive. Okay, so if you have type O blood, you have to have two O alleles because it is recessive, and that's what you need to have the recessive phenotype. So that's how I know I have type A. I have type A blood, but I'm AO because my mom is O. So the only thing she could have given me was an O. Okay, any questions about that? It's kind of cool. If you don't know your blood type, if you donate blood once you're able to, they'll tell you, which is kind of the reason why I donated blood the first time, because I just really wanted to know my blood type. Okay, so you're not going to have to do long involved problems with these. I've never seen that on the AP exam, but you do need to be familiar with this concept that there can be multiple alleles or multiple versions of a gene controlling one trait. Okay. Again, I've never seen it play out in like a free response or anything like that, but in some multiple choice questions, they may have it as like just a potential answer. So you just need to know it exists. And it's kind of just cool to know about. Okay. Last one, also pretty important, is sex-linked traits. So sex-linked traits just mean that they're either carried on the X or the Y chromosome, the sex chromosomes, okay? Much more common is having this on the X chromosome because the Y chromosome is actually really teeny tiny, which is why women are superior to men. No, I'm just kidding. But it is actually a very tiny chromosome. So the X chromosome carries a lot more genes on it than the Y chromosome does. So if something is carried on that X chromosome, we refer to it as sex-linked because it is a sex chromosome. So here's the deal with sex-linked traits. They are the disorders or the traits that they cause are much more frequent in males than females. So why is that? Because males only have one X chromosome. So whatever version of X they get is what they have. So even if it's a recessive trait, which most of these are, if they have one recessive X, then they get the recessive trait, as opposed to females, which can be carriers, meaning that they can have one recessive X and then one dominant X, which protects them from whatever it is. So really common examples of this are color blindness. Okay, so color blindness is like 8 times more likely in males than females. Hemophilia, which is a bleeding disorder, and then Duchenne's muscular dystrophy, which is an aggressive form of muscular dystrophy, which is where your muscles start to lose control of your muscles over time. So here's kind of an example of this. So in this example, we have an unaffected father for this X-linked disorder. Let's say it's color blindness. So he's unaffected, which means we know he has one dominant X, okay, because he does not have the trait. And then the mom is a carrier. She is not affected either, so she would be able to see color normally, but she has one dominant X, and then she has one recessive X that she probably doesn't even know she has. So then when this gets transferred from the parents, remember that the dad is giving an X to both of his daughters because that's why they're girls, right? Fathers are the ones who determine the gender of the baby because mom only gives an X. It's all she has to give. Dad is the one where it's a 50% chance that there's another X making a girl. There's a 50% chance that it's a Y making a boy. Okay, so dad gives his good X to both of his daughters. So right off the bat, we know that the daughters are not going to be colorblind because they have the dominant allele from dad. Okay, then one girl gets the dominant X from mom, and one gets the recessive X from mom, and therefore she's a carrier just like her mom. For the sons, it's a different story. The sons get the Y from dad, which is why they're sons. So then there's a 50% chance that they're going to get the good X from mom and not be colorblind. There's also a 50% chance that they're going to get the bad X from mom and be colorblind. So none of the daughters will be affected, but half the sons will be affected because there's a 50% chance they're going to get the dominant. There's a 50% chance that they're going to get the recessive version of the trait from their mom who's a carrier. All right. Is X-linked always recessive? It's not always recessive, but almost always it's recessive. So I will never, ever say always because there are exceptions to every rule, but for the most part, it's going to be recessive. Yep. And Madison, that actually brings up a really good point. So thanks for asking that question. If you've ever seen always in an answer choice on the AP exam, it's not accurate, okay? Because things do not always happen in a certain way, okay? And so that's like a distractor. So you shouldn't pick that answer, just so you know. All right, any other questions about sex-linked traits? Do you want to practice one? If it was dominant, what would happen? That's a great question. We would just see a really higher frequency. So both men and women would, well, both men and women would probably be equally affected, right? Because women could get it just from having one version of X, and then men could get it just from having one version of X if it were dominant. I can Google really quick. Are there any dominant sex-linked traits? It looks like there may be some. Rare. It says it's rare. So there's your answer. All right. So, okay, let's just do, well, we just did this one, so we're not going to do it. This is the example that we saw. So we know that there's a 50% chance that the son will be colorblind and a 0% chance that the daughter will be colorblind based on the example of the previous page, which you can look back to if you need extra support here. Okay, wow, we have plenty of time, so we can get into chi-square, which is honestly the bane of a lot of people's existence. So here's what I will say. I said this last year as well. College Board loves chi-square. Chi-square has not been on a free response in a while. I thought that there was going to be a chi-square on the free response last year, and there wasn't. I picture it as kind of like a ticking time bomb. So I would assume, I would prepare as though there's going to be a chi-square as one of your free response questions on this exam. Do I know that? Absolutely not. I'm not nearly important enough to know the answer to that question because I am not a College Board higher up, right? I'm a mere peasant in comparison, okay? But using my resources and looking at so many practice exams, I have this feeling that it's going to be on there. I don't know. It might not be. We don't know. Okay. Regardless, let's chat about it because I'm sure you'll be tested on it in class, even if it's not on the AP exam. Sometimes it shows up in the multiple choice as well. Okay, so why does chi-square even exist and why am I talking about it in the middle of genetics? Well, very frequently we use chi-square in order to determine if results are statistically significant. Okay, so we want to figure out usually from like a cross. Let's say we're doing a cross and we see results, okay? We see the babies of some sort of cross that we just did, okay? Are the results that we see due to chance or are they due to something else? Maybe a variable that you're testing, maybe an inheritance pattern, right? Maybe you're looking at offspring and you thought that this trait in these random rabbits that you found on your scientific adventures was just a straight Mendelian dominant recessive, but then you look at the offspring and you realize that something else is going on, okay? That's why we use chi-square. That's why statisticians use chi-square. If you ever go into research, this becomes really, 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 really important because you will use this to validate that your results are due to what you are doing and not due to the random chance of the universe, right? Because if your results are due to chance, then nobody's going to publish you, right? It's just a random coincidence, okay? So that is why chi-square matters. And again, we use it all the time in genetics to see if the results that we're seeing from perhaps a genetic cross are due to chance or something else. Any questions about that? All right, so this is actually a direct screenshot from the College Board equation sheet, which hopefully you guys know is public and available for you, so you can take a look at it. I always have it printed in my classroom so students can practice with it, because that's what you'll have on the actual exam. Do you guys know? Give me 1 second. I'm going to send you AP Biology course and exam description. I feel like this is a really important resource that a lot of students don't have. Sorry, guys, I'm like sniffling. It's gross. Okay. So this is a 224-page document that your teachers are most likely basing your course off of, and it has everything you need to know, including things to know about the exam itself, all the different units and what you should be learning in each, and then it also has this lovely equations and formulas at the end. So there's a whole section at the end that goes over an exam overview. It gives you sample exam questions. And then at the very, very end is this equation and formula sheet that you'll be seeing on your AP exam. And this is on it. Okay. So we're going to orient you to all of the different things of what this means. And then we can do some practice problems. Okay. So upper left-hand corner is the chi-square equation. Okay. So the little lovely cursive chi with the square is the chi-square variable. You do not square root it, right? It is just called chi-square, and that is what it is. You're not solving for x here. You are solving for the chi-square variable. So do not square root at any time during this process. Also, it's chi. Chi. I live in Chicago, so my students love to call it Chi because it's Chicago, Chi-town. It is chi because it's a Greek variable. And then it's the sum of, so you can see that there, that alligator mouth looking thing is a sigma, and that means the sum of the observed results minus the expected results squared over the expected results. And all of that sounds really confusing, but it's not once we practice it, I promise. Okay. And then we have this chi-squared table that we're going to use in order to determine if our results are statistically significant or not. So Madison, exactly, you do not have to memorize any of the formulas on the sheet if you know the variables. And actually, this is a direct snip from the sheet. So the variables are outlined there for you on the left. And it tells you what this term degrees of freedom mean as well. But I'm going to go over that for you in a second too. So all the equations are given to you. You do not need to waste your time memorizing any of this stuff. You just need to know how to apply it. And that's kind of the entire AP exam these days. It used to be a ton, like when I took AP Bio way back in the day in 2007. It was a ton of memorization, and now it's really all application. So they've changed it a bunch. So no, you do not need to memorize. Okay, so here's what all this mumbo jumbo means. All right, so chi-squared test we already talked about. It's a test to determine whether results are significantly different from an expected value, and it's statistical significance, which is what you need in the scientific community. Another thing that we're going to need to write out that you frequently write out for these problems is a null hypothesis. And a null hypothesis assumes that the results or outcomes are due to chance, okay? That they're not due to a variable or due to something else. So let's say you're testing whether fertilizer helps increase plant growth. Your null hypothesis would be that any difference in plant growth is due to chance and not due to the fertilizer and what you're adding, okay? And then your goal is to disprove that and say that, oh, there is a difference because the fertilizer is working, okay? So null, think of null as meaning like nothing. This is if nothing is influencing anything, and this is what you're trying to disprove if you're trying to prove that something has an effect on something else. Okay, it's kind of a weird concept, but it's kind of what we're targeting. Then there's this other term called degrees of freedom. Okay, and degrees of freedom is going to help us when we need to find a specific variable on this lovely chi-squared table. You'll notice that our headers here say degrees of freedom. What does it mean? Well, it tells you right here, degrees of freedom are equal to the number of distinct possible outcomes of a situation minus 1, okay? So really quick, easy way to describe this. Let's say we're looking at kittens, and we can have black kittens or we can have white kittens. 2 possible outcomes, 1 degree of freedom, 2 minus 1, okay? Let's say we're looking at flowers, and we can have red, pink, or white flowers because it's incompletely dominant. 3 possible outcomes, 2 degrees of freedom. okay? 3 minus 1. Okay, so that's how we find degrees of freedom, and you'll need to do that in order to locate the correct column to use on this critical value table, okay? All of this is a lot. I should have warned you. All of these definitions are a lot. They're really good to have in your back pocket though, because we're going to do a practice problem and all of this will start to make more sense, okay? So just bear with me. I know it's kind of confusing. It might be a late Wednesday night. And you may not have learned this before, in which case this can be very, very confusing, okay? So degrees of freedom are going to help us locate a critical value. And a critical value is a value from the chi-squared table, which is on the previous slide, which I'll show you again in a second, that the calculated chi-square is compared to. Okay. So all this means is we are going to use our observed results and our expected results. So what we expected to happen and what we actually saw happen. We're going to do some mathematical magic, although I just combined the two and I liked that, in order to find this variable called chi-square. It is going to be a number, okay? And we will calculate one in a second, I promise, okay? So we are going to see a number. We are going to compare that number to one of these critical values inside of this table. So let's say we're looking at cats again, and they can be black or they can be white. We have 1 degree of freedom because we have 2 possible outcomes. So the number we're comparing our chi-square value to is 3.84. Now, I don't want to get into too much detail here because this is already super confusing and this is like college level statistics, but you are going to use the p-value of 0.05 unless otherwise instructed, okay? What do these values actually mean? I will give you a brief explanation, but a p-value of 0.05 means that some statistician somewhere with way too much time on their hands said that if your results are proven with the numbers in this row, then there's only a 5% chance that they happened due to chance. AKA, we are 95% sure that your results are statistically significant, okay? If you use this row, we are 99% sure that your results are statistically significant, okay? There's only a 1% chance that we are wrong, okay? But we are okay with an A. We don't need an A+. 95% is good enough. So unless otherwise specified, you can use this row. And again, if you forget what p-values mean, that's fine. You don't need to resuscitate it. You don't really need to know. You just need to know that you should use this row of 0.05. And if that's all you remember from what I just said, I am totally okay with that. All right, so we've calculated a number, a chi-squared value. Then we found this critical value in this table. So let's say we're looking at the cats again. The value that we're comparing it to is 3.84. And we want to see if the number we calculated is greater than or less than this number. And again, when we do a practice, it will make so much more sense, okay? The last thing that we need to do is figure out what it all means. So if the value that we calculate is greater than the critical value in the table, so in my example, 3.84, then the null hypothesis is rejected, which means the results are not due to chance. We have a statistical significance, okay? If it is less than the critical value, then the null is not rejected, meaning the results are probably due to chance, okay? That's all I'm going to say on the matter for now, because I want to do a practice problem and then revisit this when it makes more sense. Hopefully that's okay with all of you. Okay, here we go. We're going to go back to bit paper really quick. Thank you guys for being so patient as we bounce back and forth to all these different topics. Hopefully you're finding some things helpful tonight. Okay, so let's set up a chi-square. So I'm going to make up a problem for us, and let's stick with cats since we've been talking about them. So we're looking at cats, which I actually don't really like cats, but that's okay. Okay, and let's say we are looking at cats where black coat is dominant. Okay, so we're going to practice some genetics, and we're also going to practice chi-square. And the white coat is recessive. Okay, and let's say we are crossing. What should we cross? Let's say we're crossing a heterozygous cat with a homozygous recessive cat, okay? And that's the cross that we're doing, and we see some results. We see some kittens, and we want to know, are these results due to chance or due to something else, okay? So the first thing that we need to do is figure out what the heck we would expect from this cross. How do we know what to expect from this cross? Well, we need to do a Punnett square. Okay, so I'm going to draw a little Punnett square over here to get my expected results. All right, and this is the easy part. I don't know why my pen is being so crappy. This tab's, okay, I get it. It's telling me that this tab's content is being shared, and I know that, so I don't know why it's being such a pain in the butt. Okay, well, these are kind of messed up, but hopefully you get the idea. You at least know that we're going to expect half of our offspring to be black and half to be white. Okay? Everyone see that so far? Hopefully we're good, because we practice stuff with that. Okay, great. Well, now that we know that, we're going to see what we actually observe. So we go visit these kittens, and here's what we see. We see 7 black kittens, and we see 5 white kittens. I'm like, oh my gosh, look, how cute. You know, kittens, when they're born, they don't open their eyes for a while. Same with puppies. So cute. So we see 7 little black kittens and 5 little white kittens. And now we're asking ourselves, are these results significant? Like, is something going on here with these cats? Or is it okay that we have 2 more black cats than white cats, even though we expected an even split? We can actually prove whether it is okay or not that we're seeing these results by doing a chi-square, okay, and seeing if the results are significant or if they're just due to chance. Because we know things don't usually work out perfectly in genetics, so it might just be that this is due to chance. So here's what we're going to do. And this table would be given to you, but you're going to have a chi-square table, okay, that's going to ask you, so you're going to have your phenotypes, black and white, okay. You're going to have your observed. So I'm going to put O for observed. That's what we actually see. Then we have what we expect. And then we're going to do the math. And remember that you already have this equation on your equation sheet. And we're going to do the math in order to find our chi-square value. Okay, so this table would be provided for you if it was on a free response question. So don't worry about having to create it yourself, but this is what it would look like. So you're just going to go ahead and plug in your numbers. So it shouldn't be too hard. So what did we observe? We observed 7 black kittens and 5 white kittens for a total of 12 kittens. We expected half black and half white, okay? So what we expected then was 6 and 6, okay? So you always have to make your expected match however many organisms you're dealing with. So we have 12 total in each column. We still have 12 kittens. We didn't delete any or add any, okay? Everything makes sense with observed and expected so far? Okay, so now we just do the math, and all of this lead up is the hard part. The actual mathematics of the matter is pretty easy, okay? So the math just involves doing your observed minus your expected. So for the first one, it would be 7 minus 6, which is 1, squared, which is still 1, divided by 6. So it's 1/6, which is it 0? No, that's 1/8. What is that as a decimal? I should know this. My goodness, 0.17. Okay, so that's what we get for our first. This is we're working towards our chi-square value. Okay. As a reminder, this is just the chi-square equation, which you have on that table. So if you're looking at the slides, you can go back and be looking at that while you're looking at this as well. Okay. All right. So then for our bottom result, we have observed minus expected. So 5 minus 6, or -1, squared, which is 1, divided by 6. So we actually get the same number, 0.17. Okay. All right, so then going back, and I'm not going to go back and switch my screen, but if you remember on the screen in our formula, we had that alligator mouth by this equation, okay? And that means the sum of. So we're adding all of our little chi-square values up to one total value. So we got to add these numbers up, okay? So we get 0.17 0.34 as our chi-squared value. Okay, so chi-square is equal to 0.34. Okay, any questions on how I found that before we move on and figure out what it means? Madison, great question. It's not negative 0.17. You will never get a negative number because the top difference is squared. Yep, absolutely. So that's always going to be the case. You'll never have a negative number because you're always going to be squaring the difference. Okay, so if you have the same amount above and below, you're going to get the same chi-squared value for that. All right, great question. Okay, so we got our chi-squared value. Now what? Why do we care? Okay, well, we need to compare this to our critical value. So how many degrees of freedom do we have here? Well, we have two possible phenotypes, so we have 1 degree of freedom. Okay, so degrees of freedom equals 1. Okay, so we're going to go back and look on our table. Okay, so I'll switch this for us just because I think it's important enough. Sorry for the little roller coaster ride. Hopefully this doesn't take too long. We're going to go back and look at our table. We have 1 degree of freedom. Remember, we can always use this top p-value. So our critical value is 3.84. So that's what we're comparing it to. Okay, so we're comparing this to our critical value of 3.84. So how does our chi-square value compare? Well, 0.34 is less than 3.84. Okay, so what does that mean then? Well, if we go back and look at our notes, you will see that it means our results are due to chance. Okay, so our number is smaller than the chi-square or than the critical value. Okay, our chi-square value is less than the critical value, which means our null hypothesis is not rejected or our null hypothesis is accepted, which means the results are due to chance. Now, this should make sense mathematically. Okay, we are calculating the difference between observed and expected, O minus E squared, okay, divided by expected. But we're calculating the difference between what we observe and what we expected to see. If that number is really big, it means that there was a big difference between what we observed and what we expected to see. That probably means that these results are not due to chance. They're due to something else. Okay, if that number is really small, it's probably because the difference between what we observed and what we expected was pretty small, and therefore the results are probably due to chance, like what we saw here. Okay, so that's a good way to remember what it means when your value is greater versus when your value is less than. Remember, you're just calculating a difference. Okay, so if the difference is very, very large, it's because something else is at play here, right? There's another variable that's intercepting and creating change, okay? If the number is very, very small, it's smaller than your critical value, it's because the difference was not big enough to be due to anything but chance, the random nature of the universe, if you will, okay? All right, so that's chi-square in a nutshell. I would love to do more practice, but unfortunately, we don't have time. Oh my gosh, Madison, bless you. I'm so glad that you're tuning into live sessions. Please let us know if you need any extra assistance, okay? But for everyone else, I hope this was somewhat helpful. Again, I wish I could go into more detail, but we're just going to run out of time. We'll probably do more reviews like this, especially closer to the AP exam when it's crunch time. So stay tuned. And then you can also Google practice problems because there are a ton of them out there. But most importantly, thank you for joining us tonight. Remember that next week we're getting into DNA transcription and translation, and then we're getting into gene expression. So all really, really important things. So definitely stay tuned. And we really appreciate having you guys. So have a great night. Thank you so much for coming.